Hello again, everybody. I'm Ryan Donnelly from the School of Pharmacy at Queen's University in Belfast, and I'm delighted to be joined for the latest in our series of calls with the Luminary with Professor Khuloud Al-Jamal. Um, you're very welcome, Khuloud, and it's great to see you.
Hello, Ryan. Nice to see you, and thank you for the invitation for this great opportunity talking to the CRS community.
Thank you very much, Khuloud. So Khuloud is Professor in Drug Delivery and Nanomedicine, and also the Head of Medicines Development at King's College in London. And she has a long and successful track record in the nanomedicine area in particular. So Khuloud, I'm first going to ask you, how did you get into the field? Because you're a pharmacist by training. How did you decide that you were going to focus on nanomedicine research?
So serendipity is the answer, Ryan. So I started my journey back in the 2000 as a PhD student from Jordan, where I decided I would like to pursue my PhD studies either in the US or UK. I equally tried both in both countries, and the destiny took me to the School of Pharmacy at the University of London. At that time, Professor Alexander Florence was my mentor. So I still remember these days when I went to see him the first time, and he was very excited about a new class of nanocarriers called dendrimers. So dendrimers are, in Greek, they mean a tree. So really, they are a tiny, tiny hyperbranched polymers, and many people still argue whether these are nanocarriers or not, but they are certainly nanoparticulate material. What was exciting about dendrimers was their size, because you could make them as small as 5 nanometers in size. You could grow them in different generations, so you could have a second, third, fourth, and so on generations. So it was actually my first exposure to what's called at that time not nanomedicine, but was drug delivery. And a very useful definition that at that time Sandy told me, drug delivery is simply altering the pharmacokinetic of a drug. So you're changing the fate of a drug by putting it into a carrier that takes it somewhere else in the body where you really want it to go. And that may have a consequence, which is improving the bioavailability of the drug at the site of action and reducing its off-target effect. So this was my first exposure to what we call nanomedicine these days, Ryan.
Yeah, and whenever you then started into your PhD, was it something that you really then got interested in and decided that you would like to have this as an academic career and develop your own group, as opposed to, for example, taking up a role in industry? Because coming from such a world-renowned group, you would have had many options for the next step in your career after your PhD.
So, Ryan, I've always been passionate about teaching and academia. So even before undertaking my PhD, I worked as a teaching assistant for 2 years in Jordan. So teaching and education is my passion. My colleagues now tell me that I actually was teaching them while we were doing our PhD study. So I think it's something in my blood without noticing. Now, what was intriguing is at the time of doing research, I never thought I want to be a professor, or I didn't think what's next, as much as I was enjoying and doing well what I was doing, and really doing it with passion to the best I could. After finishing my PhD, I have an interesting story where I did my pre-registration training post-PhD. And it was a time where I was doing my write-up for the thesis and doing my 9 to 5 hospital training at that time at UCLH, a very well-known hospital in London. So it was a tough year because you have to manage your time, study for your pre-reg exam, write up, and your supervisor is chasing you to submit by a deadline. So I managed that, and probably I would say that year of pre-reg training made me miss academia, made me know that is the right place for me. And I was lucky to have a postdoctoral fellowship lined up for me, which I started straight away after my pre-reg year.
Excellent. Khuloud, you mentioned Professor Florence there, and I think that many people will know him as a great mentor of PhD students, postdocs, but also of academics. So maybe you could tell us a little bit about how important that mentorship was to you, and how that's influenced how you now mentor people who work in your group.
So I say to my students and to people in general, people have different style. There isn't right or wrong. So, for example, I had a certain a way of running the lab when I was a student, which was completely different from what I had during my postdoctoral training, which I equally found useful. So I'll tell you later about my journey of the postdoctoral fellowship, but what I can tell you, what I've learned from Sandy is how to explore your own ideas and actually be observant to details. I do remember one time I went to him and I said to him, my dendrimers are fluorescing when I put them under the UV light. And it was a cheeky way of putting my dendrimers in a dialysis bag and seeing, does it really come out of the bag? So I would have put it under the gel dock, which at that time was a kind of a transparent window, and I see this blue light. And my dendrimers had no chromophore. So then, the first thing he did was going onto the Google and put fluorescent dendrimers. And there was one paper where it was an unexplained phenomenon. But what you can see is when you have a concentrated electron donors, amines, around a core, you sort of have some sort of energy transfer, which till now it is not explained. But then, we wrote a couple of papers on that. PhD students follow that on to track dendrimers in cells just by intrinsic fluorescence. And what I learned was no matter how trivial or simple the observation was, follow it. This is how research grow and generate. I learned things from him like sometimes the students need to talk to you in a time when you're busy. So I still remember when one day he had a very important reception where I actually went on and called him and I said, can I talk to you? And I had, to me at that time was a problem of the world that I could not reproduce my experiment. Mm-hmm. So actually, you know what? He left the crowd, he came and spoke to me, and he gave me an advice, which I still remember, start a new direction or and so on. So this is something that I will never forget. Listen to the students, talk to them, and see how they see things, because actually they may be right. They are the one who are doing the experiments. So certainly certain things. On the other hand, I did my postdoctoral training in the Costa-Rica lab, where I've learned something different things, and me is a combination of all the experiences I had, and I owe all of them a lot.
Yeah, and whenever you moved then to do your postdoctoral fellowship, Khuloud, were you making a big departure in terms of research? Was it a very different type of research you were doing? And how did you choose that was the postdoctoral fellowship that you were going to do? Why was that the particular area you focused on?
So this is a very interesting question, Ryan, because my postdoctoral training was actually a follow-up from a failed experiment. Mm. So in the final stage of my PhD, I had this hypothesis where actually I would like to use dendrimers, which are highly positively charged, to deliver anionic macromolecules such as heparin. So I made these, I started mixing them, and they were forming these very nice, precise, what we call polyplexes that they were fantastic in terms of size and positive charge and so on. When I did my in vivo biodistribution study, it was a disaster because actually we could see no improvement in absorption whatsoever. In fact, if anything, it neutralizes the activity of heparin. And we published that in a journal. It's called Results in Pharm.Sci, and the results were negative, but actually there were results, right? So then I took that as a basis for my postdoctoral fellowship, where I wanted to investigate if dendrimers can bind heparin so strongly, could we use them in vivo to bind heparin and act as anti-angiogenic agents? And that was the basis of my first fellowship, where I spent 2 years exploring. And actually, if anything, that period was a training exercise for me to start my preclinical and animal studies. So I developed animal models, I developed spheroid models and intravital microscopy to explore and validate my hypothesis, which was then published in PNAS. Now, you can see that it was a complete switch from chemical synthesis and physical-chemical characterization to preclinical translation and in vivo imaging. And this is where I got to learn all the models that were basically crucial to assess nanomedicines. So when I took this further, so after that, I undertook another postdoctoral fellowship. So my postdoctoral fellowship was for a 5-year period, but it prepared me when I started my lectureship to actually knew what I wanted to continue with, apply this into specific disease, which was cancer, and put in my strong grants application. So what I advise PhD graduates is postdoctoral training is very important. And this is actually when you consolidate the knowledge you gain from your PhD, work with peers, publish a profile, and strong papers. So I slowly transitioned into applying my knowledge into specific diseases, more specifically cancer and brain diseases. And actually now we're working more on specific therapeutics and specific disease models that we would like to treat.
And so what is your lab focusing on at the moment? And what are their main research areas? Because I know that you have still quite a broad portfolio of things that your group are working on at present.
Yeah. So we're interested in terms of nanocarriers. We have a toolbox of different materials because we believe each disease, each therapeutic cargo you want to deliver require a special designer nanocarrier. So it's not one size fits all. And we use this knowledge to pick the right carrier to deliver the right molecule. So we start with the problem. So at King's College London, if I'm sure you're aware, we have very strong links with clinicians, and clinicians have a specific problem that they would like to solve. For example, a microRNA that they know if you deliver to the heart, it helps cardiac regeneration, or something that if you deliver to the brain, this is what you're going to get. So we start with a problem. We pick the drug that we have, we would like to deliver. And we choose the right delivery system. We perform the pharmacokinetics analysis to ensure that we are delivering the drug where it has to be delivered, and then perform things like pharmacodynamics and so on. So back to your question, Ryan, I'm interested in cancer, I'm interested in brain diseases, I'm interested in novel delivery routes. For example, now, for the brain, we had in the past exploited the systemic delivery to the brain, and we know more about the blood-brain barrier, how to overcome it, what are the characteristics of nanoparticles that are desired. So now we're exploring nose-to-brain delivery. In terms of therapeutic cargoes, we are coping with the new advances in the field. So, for example, post-success in terms of siRNA delivery. We're working now at mRNA delivery and in vivo gene editing using mRNA delivery approach. So I would call, a third generation advances in the field. We are interested in combining immunology with the diseases. So we know a lot about cancer, but with the successes of Nobel Prize winners in immune checkpoint inhibitors, there is more to know about why previous treatments, which we injected, worked or did not work. So combining science of another discipline to what we do I think is very crucial. And that's what we're trying to do, combinatory, whether that is a combinatory field or combinatory route of administration plus a delivery system, so towards complexity, Ryan.
Yeah, and I think that's probably been one of the main reasons why your group continues to be so successful, Khuloud, because you are employing diverse methods and also in taking learnings from several different spaces and applying those then to targeting a drug to a certain way or certain place, or understanding why a nanomedicine is behaving in a certain fashion. And it's obvious that you continue to innovate in that space. And I know that it's obviously a very busy area of research, but also there are a lot of challenges in this area. So I'm wondering, Khuloud, as an expert in this field and one of the leading drug delivery specialists in this field, what do you see as being the main challenges in this space at present?
So I think the challenge from what I see, for example, there's so much competition around. Probably a lot of people are doing the same thing. Mm-hmm. And for example, in terms of what we do in terms of patent applications, taking this by an industrial partner, there's a lot of similarities, and unfortunately some people are not doing it very well. Mm-hmm. But yeah, I mean, there are a lot of publications that are misleading in our field, and you try to reproduce them, they're not reproducible. So the challenge I would say is I think we need to pick the right diseases to treat with nanotechnology. For example, I'm very interested in brain cancer. I'm interested in rare diseases of motor neuron disease, which are currently have no treatment. So really any incremental improvement on existing therapeutics. Why would you give a complex system if you could treat it with a simple drug, right? So there has to be a strong reason. And clearly there are many diseases that have failed the conventional treatments, and we need to find new approaches to treat them. So I know there are challenges, yet there are lots of hopes that we can exploit.
Exactly. And what about translation, Khuloud? Because I suppose this has been one of the things that have been at the center of many discussions at the Controlled Release Society annual meeting about translation of nanomedicines and the difficulties in taking something that looks really promising and translating that into a medicine that will actually make a meaningful difference for the quality of life of patients. Is there anything that we can do to improve translation?
Yeah, I think standardization in how we characterize these nanomaterials and reproducibility. Each student who leave my lab, I ask them to write an SOP for how this work can be done, because sometimes you can't reproduce the data simply because the information is not precise enough. So what I would really advise people is to document and have standardized protocol, even within their own lab, that then can be shared between the community. In terms of translation, I mean, very good example now, we have these COVID vaccines, and the issue of storage at -80 has been a problem. So one way is to have a proof of concept efficacy, but then if this is good, think of how this can be made available and economical to processes such as distribution, storage, and so on. So there is a lot of challenge in that. Probably I would say scale up and being able to have batch to batch similarities, especially when it comes to biomolecules and biotherapeutics. And cost. These things are not cheap and quite expensive. But if we look at the cost of failed treatments, for example, in the NHS, you can see sometimes you could still achieve the same effect if you have. What's important is us, right? Human beings, and getting the right treatments for them. So we have to appreciate that there might be cost implications in the beginning, but then everything starts expensive, and slowly we can find ways around it.
Exactly. Khuloud, you've had a very rapid rise in your career, you know, and I'm wondering if you have any advice for any scientists who are doing a PhD, in terms of, you know, how, you know, you can progress in the academic space, and, you know, what have you, you know, particularly enjoyed about your time in academia?
So I enjoy new ideas and really intellectual discussions with colleagues, students. And I think I would say, I mean, luck is one thing, Brian, but if anything, I would say working hard and enjoying what you're doing. Do it with the best way you could. People have different abilities, and it is. I think the best way is to know where your strengths are and exploit them further. I feel also publications are quite important. For example, I know at the moment things have changed where quality versus quantity, but for young researchers, even if you have a small paper that could carry negative results, the exercise of writing the paper, getting it reviewed, how to rebuttal your paper is very important. So for young generation, I would tell them, write up your work, publish it, no matter, you know, how big or small the paper could be, as long as it is good work, is a key.
Yeah, of course. And one of the things that, you know, you've done very well, in addition to the funding and achievements from your group and publishing very widely in quality journals, is to present your work very widely across the world. And you have several visiting professorships in Italy and in China. And you also have presented your work at numerous conferences, including the conferences of the Controlled Release Society, and you've had a long association with the CRS. And I'm just wondering, Khuloud, what, you know, your time as a member of CRS has meant for you, and you know, what opportunities has that created for you?
So my first CRS conference, Ryan, was in 2002 in Seoul. And it was a memorable journey that I will never forget, because I had to end up flying to 3 countries before I arrived to Seoul because of some logistical arrangements in, you know, internal flight in Italy. But after 3 days of travel, I made it to Seoul, and this was my 2002 conference. So I was, initially as a PhD student, you really don't know. You're told what to do, right? So I could see that presenting to a conference was key. I still remember the late nights we had to work to get that data to present in your poster, because you were not allowed to present some old data. And since 2002, I have been to numerous CRS conferences, and it's the conference that I would say I've learned a lot from. I got to know a lot of colleagues and friends across the world. I was recognized by some of their awards. 2 years ago, I had the CRS Young Investigator by one of the focus groups, drug delivery focus group. So I do appreciate the friendship I had with people, including yourself and many others. And it feels like my academic community and family. And I do strongly advise the students to participate with posters or with short talks, because they will be their future academic friends that they will reach out to.
Yeah. So with that positive note, I think we will conclude our discussion. Professor Khuloud Al-Jamal, thank you very much for joining us.
Thank you very much, Ryan. Thanks to the CRS community. Thanks.